Thank you, John. So thank you for having me. So let's get started. So viral helicase. So while virus replicating inside of the cell, especially RNA virus, it generates a ton of RNAs during the replication cycle. So one of the unavoidable intermediate during the process is double-stranded RNA. Without untangle this double-stranded RNA into a single strand, so actually the RNA may not serve as for its purpose, such as genomic RNA or messenger RNA. So now we kind of viral helicase comes into play. So in order to kind of unwind this double-stranded RNA, become into a single-stranded RNA. So actually, in the notion on the left side, this is kind of cryo-EM structure that's solved by the Rockefeller group. So not only for the RDRP, in this case, the SARS-CoV-2, but also the other accessory proteins are actually kind of modeled within this, the replicase complex. So not only for the other small protein, but also actually there are two copies of the viral helicases forms with the really kind of nice replicase complex, which actually underscores the importance of the viral helicase during the replication cycle. So because of that, so people has perceived this kind of viral helicase as a good target for next antiviral inhibitors. However, there are not many actual realization has happened in terms of kind of developing compounds targeting viral helicase as an antiviral target. So, but these three compounds that I'm showing you here is kind of one of the rare, three of the rare example. Amenavir is actually anti-HSV helicase or primase inhibitor because this actually has been found from the phenotypic screening, and then they found the resistant mutation within this helicase and primase complex. So this is kind of one of the examples, but bottom line is there are not many viral helicase inhibitor that's for antiviral target purpose wise. So from this idea, so the Midwest AViDD Center formed some projects focused around viral helicase, and this is a project 5 we call. So I'm the leader of this project with many medicinal chemists and other people too. But this idea actually has not come from out of the blue, actually. So we have some kind of ongoing projects for more than 12 years in my lab with the collaboration with Dr. Colin Johnson and Jennifer Golden. So actually this project is for the alpha virus program. So we started with some kind of phenotypic screening cell-based assay using 350,000 compounds. Back then, it was kind of quite huge library. What ended up with being is actually one compound, quinazolinones. Out of this screening, we found EC50 for live cell virus assay was less than 1 micromole. And then we found that this is quite interesting compounds, and then we follow up with some medicinal chemistry and then everything. At the end, for now, actually, we have a kind of less than 1 nanomolar EC50 in live virus. And then also in vivo, we showed kind of 100% protection, even delayed treatment kind of thing like that. So actually, it kind of gives us a good confidence that these types of approaches work. So however, more surprising outcome actually has come from some sort of kind of mechanism of action study. Because this compound has come from some sort of phenotypic screening, we were wondering what is the actual real mechanism. When we actually found some resistant mutations, actually we found resistant mutations are mapped within NSP2 of the virus protein. In this case, NSP2 is a viral helicase. So it actually gave us some kind of justification. Inhibiting of the viral helicase could be a really good target for other RNA viruses as well. So back in 2020, when SARS-CoV-2 actually the pandemic started, we compared this SARS-CoV-2 viral helicase structure against our NSP2 VEV helicase compound, the protein structure, as you can see. So their topology is almost identically same with a little bit of a change here and there. And also not only for this SARS-CoV-2, we also found other RNA viruses encode their own viral helicase for the purpose of the unwinding of their genomic and subgenomic RNA. In this case, actually, SARS-CoV-2 and alpha virus, both of them are superfamily 1 helicase. Their translocation direction is 5' to 3', but the other superfamily 2 in this Flavivirus has a different opposite orientation. So it actually gives us some kind of starting point to how do we start this one. So as kind of so Chaim already mentioned, we started also the ultra throughput, high throughput screening. So idea is very same. So we have one end of the DNA actually quencher, the other one end was as a FAM. So once RNA helicase, or in this case as SP13 of the SARS coronavirus unwinded, we can actually gain of the fluorescence signal. This is a very simple format of the assay. We took a lot of advantage from the literature. They actually developed these types of assay using SARS-CoV-1 about 10 years ago. Nice thing about this SARS-CoV-2 helicase is actually there is only one amino acid change between SARS-1 and SARS-2 helicase. So we didn't have any problem in terms of implementing of the assay. So using this developed assay, and so we implemented this one into 1536 plate format. Usually, and then this screening was done by the UF Scripps group, starting from the 650 compounds, and go through all the funneling channel. At the end of it, we kind of have a little bit of a kind of 700 compounds showing the IC50 about the 10 micromolar level. So if you actually count the number of hits based on the IC50, and you can actually see median or some mean IC50 is around this 5 micromolar levels, and you can see some of the better compounds than the 1 micromole. And then if you just kind of get rid of all the potential pain or some sort of kind of promiscuous compounds, and also if you actually kind of do more diligence in terms of IC50 wise, you can actually get down around the 270 compounds having IC50 value less than 5 micromole. So from these compounds, we actually collected these compounds, repurchased from the scrap, the fresh material, and then we called this is antiviral helicase library, consists of around 220 compounds inside. So with these compounds in our hands, we actually ran many different assays simultaneously, tried to gain insight to which compounds they actually provide us actual antiviral activity out of this library. So throughout the talk, I'm gonna utilize some kind of 2 frontline antiviral assays. One of them is based on the live SARS-CoV-2 in A549 cells with the AC2 integrated one. So this one assay is using the CPE-based assay. So if you have antiviral compounds, it means the cell would be been protected from the cell death from the virus CPE. So as the concentration goes up, you can actually gain of the signal on the shown on the left side. The other assay is paired with the CPE assay is an log assay we call. So this is actually nanoluciferase is integrated into SARS-CoV-2 genome. So we use this log assay as a kind of surrogate for the virus replication. So in this case, we can actually see the decrease of the signal when the virus replication goes down as concentrations goes up. So by using 2 readout, one goes up, one goes down, we can actually narrow down kind of really good antiviral compounds from time to time. When we actually have a really good compounds, we actually test them in live virus yield reduction assay. This is more likely traditional assay using determine the virus titer in the presence or in the absence of compounds, and then we do the log transformation, and we calculate how much virus fold reduction achieved in the presence of the compounds. This is kind of our backbone of the assay from this talk. So now, so out of this 220 compounds show IC50 values in enzymatic assay. So we actually see about the 90% of the compound, it didn't show any types of activity on the right side of category 6. So this kind of expected, right? So we don't expect that all of the anti-helicase and compounds may be the antiviral. So, but then as you can categorize this one based on the specificity to the virus as well as some cytotoxicity, you can actually gain of some kind of category number 1 and 2 shows specific to SARS-CoV-2, specific but has a little bit of a toxicity kind of thing like that. So, and also not only for the SARS-CoV-2 in this assay, we also ran this same set of the compounds against the chikungunya. This is our counter screening because we don't want to have any compounds that actually bluntly inhibit the cell. So it can be a false positive later on. So we just wanted to get this in place in order to select the compounds. Out of this 220 compounds, we were getting down into about the 7 or 8 compounds showing IC50 in live virus assay around the kind of low micromolar level up to 30 micromolar level kind of thing. However, all of the SI value is greater than 3, or sometimes you can get to the 15. So this is kind of a very fresh result to hit compounds from the high throughput screening. And then we quickly move to the actually live virus titer reduction assay. So-called without compounds, with compounds. In this case, we ran 2 concentration, 20 and the 5 micromole. So at 5 micromole, actually, you can see the MWAC 552 actually shows a meaningful virus reduction, about a 1 node, which means 10-fold the virus titer reduction. Another good compounds out of this assay was 110 series. Actually 110 compounds shows also about a 0.7 or 0.8 log reduction in this assay. Not only for this single compounds, we also make some kind of purchase, some kind of structure-based analogs from the commercial compounds. And then out of this series, you can actually see some of them, in this case, 82, didn't show any activities, all the analogs, which means this series may not be a good thing to follow up. But as you can see here, 5210 actually shows some spectrum, which actually we wanted to see. We don't want to see any flat kind of SAR, but actually so 52 and 110 shows a nice SAR from the category, the purchased compounds. So we are very confident that these 2 series may be the good things to follow up. And then medicinal chemistry kind of synthesis effort coming along. So I'm just showing only 5 compounds from this series, but we made more than 50 or 60 compounds out of the series. So as you can see here, pleasantly, we see 2 compounds from the 52 series, actually less than 1 micromolar antiviral activity in both assays, and without any activity against the Zika or SARS-CoV chikungunya, or without any cytotoxicity. So it gives us a kind of selective index of more than 100-fold, which is pretty good from my perspective. The other series, MWAC 110 series, also have 2 compounds that are down about the 1 micromole or sub-micromolar level of EC50 in this case. So we just kind of decide to follow up these 2 series further down. And then not only for the EC50 wise, we also measure the kind of their efficacy in 2 different concentration, 10 and 20 micromole. As you can see here, so 1622 actually stood out because even 10 micromole, we see almost no virus replication happens in these cases. Out of 110 series, we found 2192 compounds that shows a really good antiviral activity in titer reduction assay. If you do the more data point here, if you, as you can see here, we see the nice dose response curve in terms of virus progeny virus titer wise, so which is pretty exciting for us. The nice thing about these 2 compounds is that it such has a kind of great virus reduction in 10 micromole without any cytotoxicity. So it allowed us to actually do some kind of genetic study out of this one. So what we did was we wanted to find some resistant virus in terms of mechanism of action study. So this is kind of very standard format. So we culture the virus in the presence of the compounds and about the 10 passages. So we start from about EC50 or EC90 concentration for each compounds. Every other passage, we increase the concentrations stepwise fashion. At the passage 10, so it's about the 10 micromole. So we did, if there is wild type wise, we are not supposed to see any virus replication. If we see this virus replication, it could be the meaning of the kind of resistance virus coming along. On the right side, I show it's kind of P0 versus P10, actually even P10. So the DMSO, we actually did a plaque assay without with the compounds, as you can see here. So virus passes number 10 in the presence of 1522 actually show the plaque, which is very strongly suggests that these are genuinely resistant to virus. Same goes with the 2192 selective compounds too. Of course, we wanted to understand where the mutation is. So we actually run two different sequencing approach. One is a population sequencing. I showed the kind of dandelion plot. So we have three circles shown here. First, the bottom one is a P1, and the second one is a P4. The last one is a P10. So if you can look at this and then see where so low versus P1 shows about the 15% mutation, and then this proportion get increased as passage goes up. Now you can see that this is the kind of populations, the sequencing. If you kind of summarize everything, 1622 passes virus population, so it's about the 75% of the population has a mutation in NSP13, 204 region. The other two mutations also found from the 2192 passage virus. One is a 186, the other one is a 479. But this does not guarantee actually these are the resistant mutants that are conferring the resistance. So what we did was actually we individually isolated this mutant virus, more about the 10 or so from each one. And then as you can see here, all of the mutant virus we isolate actually has two mutations commonly found. One is within the spike protein. So the other one is one of the NSP13. All of them has this common mutation. So spike protein actually makes sense because with this virus produced within viral cells, now we are passing these cells in the A549 cells. I think this is kind of gain of the kind of new type of attachment factor kind of things. Like this actually same mutation was found within DMSO treated to control the population as well. And then noticeably, actually, you can find that we were very pleased to see two clones actually has no other mutation, but only two, which means, so we can actually remove all the alternative explanation, but we can actually focus on this NSP13 mutation, whether this mutation is actually mutation conferring the resistance or not. Same goes with the 2192. The one difference is we can actually find the two different loci. One, however, the same story goes here. We found several 2 or 3 mutant virus clone that has only those two mutations other than this is just kind of clean background. So in order to kind of prove this mutation is actually conferring the resistance, so we actually tested this isolate against their own, antivirus. As a control, we tested this susceptibility to the remdesivir. As you can see, wild type shows 0.1 micromolar EC50, but we don't see much of a changes in our resistant virus, mutant virus per se. So we see barely almost no changes in EC50. But in case of the 1622 passage virus here, 204, actually you can see about the 20-fold increase in EC50. And in the case of this 2192, actually you can see about the 6 or the 18-fold increase in their EC50. So which actually proves that this NSP13 may be the viral target, because we don't see any other mutation other than this plus some mutation within the spike protein. So I think this is kind of very strongly argue that our compound actually interacted with some NSP13. If you actually map out this mutation in the context of the structural biology perspective, so this 204 mutation is happened to be 1B domain here, and the other two mutation we found from the 2192, actually one of them is still 1B, the other one is a RAC2 domain. So one interesting fact about this 204 is actually, so we found that this actually 204 is engaged in a salt bridge between the 1B domain and the RAC2 domain. So if you have a mutation in this aspartic acid glycine, you may not see these types of salt bridges. So we currently think that this kind of mutation actually allow this 1B domain more slippery, so it actually kind of have some sort of effect on the binding of the compounds to the compounds. That's kind of our current hypothesis. if you actually focus on then how indeed this compounds work, so this is kind of one of the conundrum that I wanted to bring your attention. So when I presented this cryo-EM structure, one of the kind of shocking things to my mind, at least, was that because this NSP13 is engaged with some their parental strain, template strain, not the synthesized strain. So one of the conundrum is NSP12 wanted to go this from 5' to 3', but at the same time, NSP13 also have the same directionality. So if they are within the same complex, how this can happen? So because of this puzzling fact, so actually the Rockefeller group actually proposes some sort of kind of RT backtrack complex, which means just in general, when the RNA synthesis happens, this 1B domain does not engage with the RNA. So it just kind of, we say this is open form. So in the case, just kind of keep RNA is making because of the RDRP proportions of it. But, and then, however, when there some sort of mismatch happens, that's, and 1B domain actually kind of turn, swivel form, and then actually it allows the RNA engaged with the 1B, the group. So now actually this can go back in order to cleave that errors and then make some sort of kind of backtrack formation. This is kind of current hypothesis in the field, how this entire complex works in under the assumption of this tug of war. So we think so our compounds probably has to do with some of the steps. So still we are actively working on how actually impact of our compounds in this transition state. So this is kind of as of now. So now we are kind of moving toward into more virology question. Indeed, how these types of compounds affect virus replication. So this is more likely more than anti-helicase. Now we are talking about how it makes an antiviral. So this is kind of pretty standard form of say time of addition study. Now we are using ICSAR. So this is kind of BSO2 virus because of complementation system of the ORF3 and E genes. If you add the virus to the cell, and you can actually start the seeing of this reporter signal around the 6 to 8 hours, which is probably meaning of the subgenomic RNA synthesis at this time point. If you add the compounds around 4, you don't see much of the activity, but if you start adding compound to 6 hours, you kind of see no effect at all. So which means our compounds probably working around 2 to 4 hours during the replication cycles of it. And if you measure the viral protein, actually you can get the same idea how it works. But if you actually look at the viral RNA synthesis, you actually get some different the landscape here. So this is kind of without any treatment. You can actually see the nice increase of the viral RNA. So this is a real-time PCR data targeting the viral NP gene as our readout. So this is nicely correlated with the kind of model proposed by the Dr. Saruki's group. You can actually see that some kind of genomic RNA synthesis early replication cycle. In the middle of it, you can see a lot of messenger RNA synthesis becoming the protein. And then, however, if you add a compound in the system, what you're seeing is quite some puzzling too. 2192 compounds, it kind of makes sense because everything goes down. So which means probably every single step of the RNA synthesis goes down. But the 1622, which actually has a higher activity in terms of antiviral activity, did not produce much of the decrease in RNA synthesis. So this is kind of very puzzling for us, so how it this happened. But our current notion is that probably the 1622 has to do with work on the subgenomic RNA synthesis inhibition, but the 2192 may inhibit all the process or all the infections. That's kind of what we are seeing. So what I'm saying is probably this much of the extent of the RNA may be coming from this genomic RNA synthesis. This much of a deficit may be coming from the messenger RNA cells. That's kind of our current model. One other thing I was just wanted to briefly touch upon is kind of what about anti-helicase versus antiviral? So if in an ideal world, you might want to see some kind of nice correlation between unwinding activity versus cell-based activity. So you can actually see that some kind of clusters of our hit compounds. I mean, this is by design because we selected the compounds showing the nice IC50 and the nice AC50 at the same time together. But if you kind of keep following all the analog compounds, all of the series, you can see it's kind of there all over the places. We don't have any more good correlation between two assays. So you can explain it because of kind of some kind of solubility issues or some kind of some exposure issues or whatever. You can just kind of name everything. But what I'm saying is that, for example, 52 series, many of the compounds shows a really good viral activity in this case, but it does not show much of the kind of anti-enzyme activity. So this is kind of really kind of puzzling still. I have some sort of kind of speculation that why this happens. So one of the notion is that whenever we do the high throughput screening or small biochemical assay, we typically add only one component, which in this case NSP13. But if you think about this one as a context of the kind of virus replication cycle, this is one of the components of this big thing. So movement or biological activity from the single protein versus within a complex may be different. So the other things also I need to talk about is biochemistry in vitro may not be truly represent what's happening inside of the cell. So NSP13 in this case is for example, this Dr. Kevin Ronnie actually said, they measured how much ATP consumed within in vitro study. They said about 800 ATP per second per molecule. If this is actually happening inside of cell, this does not hold the truth, what's happening inside of cell. Secondly, if you actually express the NSP13 in bacteria, it does not have the ion sulfur cluster in the terminal domain. So there is some nuanced differences in here. But still, whenever we do a large scaling of the high throughput screening or some enzymatic assay, we just tend to forget about this one. Another weird thing is SARS-CoV-2 is that although this is double-strand RNA virus, it has a higher processability affinity to double-strand DNA. So this is kind of one of the examples that I'm bringing to. Another thing is the dynamics, because when you have a single protein, so you don't see this types of dynamic the interaction between the proteins, but this only happens whenever you have a multiple protein as a complex. Another thought is kind of biological issues, such as where this replication complex was in. As Chaim said, this is kind of within the double membrane vesicles, which is quite different environment than just kind of in vitro system. And also how much is enough to show the antibody effect. So IC50 may not actually correlate with the EC50 or virus titer reduction, because even though you inhibit, say, 90% of enzymatic activity, but 10% is still produce a viral protein that could be related to the virus replication. So it's kind of a lot of things we have to think about. I think so why the protease inhibitor was very successful is probably protease works as a monomer dimer as its own. So it can be very much translatable, but in the case of a virus replicase complex, it could be a different story. That's kind of what I'm trying to say. In summary, so probably I already time's up. So ultra high throughput screening compounds, we got about 300 compounds with good anti-NSP13 activity. So out of this, we found 7 compound series that come from this specific antiviral activity. So we are actively following up with the 2 series compounds. And then so resistant mutation study actually identified good evidence that accompany the resistance within NSP13. And then so we think NSP13 is not just, it's probably more than just one unwinding, because depending on the compounds that actually we can see the different phenotypic effect in what types of species of RNA is being synthesized. So we think NSP13 may have a crucial role in terms of virus replication as well, the controlling mechanism as well. So of course, this is a huge work throughout the whole our Midwest AViDD Center. So I cannot name everyone, but most of the high throughput screening was done the UF Scripps team, and the medicinal chemistry was done by the chemistry cohort of Bannister, and the computational, all the structural biology was done San Diego, Berkeley, and many members too. So with that, I'll be happy to take any question, but I feel like I used all the time that I gave. Thank you.